Marina ma'am?
Yes.
Ma'am, shall we start now?
Go for it.
Yeah, thank you, ma'am. Helen?
Yes, ma'am. A very pleasant morning to one and all gathered here. On this glorious day, I feel privileged to extend my welcome to all those present here for the second session of the International Webinar Series BIOFIESTA 2023, organized by the Department of Biotechnology, Sree College, in association with the Internal Quality Assurance Cell IQAC and with the financial support of the SMART scheme. Peace on outside comes from knowing God on the inside. Let's bow our head and put ourselves in the holiness of God Almighty. I invite Ms. Saurasena for the prayer.
தெய்வமே காத்து கொள்கங்கு കൈവിടാതിങ്ങു ഞങ്ങളെ നാവികന്നീ ഭവാപ്തിക്കോ രാവിവൻ വസ്ത്രാദിമുട്ടാതെ തന്നു രക്ഷിച്ചു ഞങ്ങളെ ധന്യരാക്കുന്ന നീയുന്നു തന്നെ ഞങ്ങൾക്ക് തമ്പുരാൻ.
Thank you. Moving forward, I welcome Ms. Layamal for the welcome speech.
A warm welcome to one and all gathered here. The galaxy of intellectuals, Your Excellency invited eminent speaker, guests, professors, and dear friends. It is a matter of great pleasure and pride for me to welcome you all to the second section of International Webinar Series BIOFIESTA 2023, organized by the Department of Biotechnology, Sree College, in association with the Internal Quality Assurance Cell and with financial support of SMART scheme. Today's webinar scientific section mainly focuses on advances in neurodegenerative diseases. Neurodegenerative disorder encompasses a wide range of conditions that result from progressive damage to cells and nervous system connections that are essential for mobility, coordination, strength, sensation, and cognition. Neurodegenerative disease affects millions of people worldwide. Scientists recognize the combination of a person's genes and environment contributes to the risk of developing a neurodegenerative disease. Clinical research entails examining exposures that may have occurred before a disease diagnosis and understanding their effects. The mission of neurodegenerative disease research is to promote and conduct multidisciplinary clinical and basic research to increase the understanding of cause and mechanism leading to brain dysfunction and degeneration in neurodegenerative diseases. So in today's scientific section, we are focusing on a very relevant title, Advances in Neurodegenerative Diseases. Before moving into the depth of scientific section, let me get done with my duty to welcome you all. Some stars shine at night and others shine in bright daylight, and it's our pleasure to welcome one such star, Dr. Merina T. Varghese, as our resource person of the day. It's double sweet to have Dr. Merina as our resource person because our HRD, Mrs. Sujya Lakshmi ma'am, and Dr. Merina were also college batchmates. Both have completed their masters in biotechnology from the Department of Biotechnology, Cochin University of Science and Technology, Kerala. We welcome you, ma'am. On this occasion, I have immense pleasure in extending warm welcome to all the faculty members who work so hard to make this a successful program. Welcome you all, dear teachers. Last but not least, I welcome all the faculties, guests, professors, students, and dear colleagues who are here to attend the webinar. On behalf of the Department of Biotechnology, Sree College, I welcome you all. Thank you.
Thank you, Layamal. Next, I would like to invite Ms. Madangi Verma to give an introduction about the eminent speaker of today's event, Dr. Merina T. Varghese.
It is a matter of great pleasure and pride for me to welcome our guest of honor, the eminent speaker of today's event, Dr. Merina T. Varghese. A distinguished scientist, an excellent professor, a seasoned scholar, and one of the eminent scientists of United States of America with an international reputation. She is a woman of deep wisdom and vast experience in the areas of spatial proteomics and cellular pathology in brain aging and disease, neuron morphology in neuropsychiatric disease, cellular alterations in neuropsychiatric disease, and mitochondrial function and protein aggregation in neurodegeneration. She completed her Bachelor of Science in Zoology 1997 to 2000 from St. Teresa's College, Mahatma Gandhi University, Kottayam, India with University First Rank. She completed her Masters in Biotechnology from Cochin University of Science and Technology, Kochi, and secured University First Rank. She completed her doctoral studies in neuroscience in the years 2003 to 2008 from the Indian Institute of Chemical Biology, Jadavpur University, Kolkata, India, on the title Mitochondrial Dysfunction in Parkinson's Disease. She started her research career as a summer research fellow in 2000 at Jawaharlal Nehru Centre for Advanced Scientific Research, Bangalore, India, as a project trainee in 2002 at Rajiv Gandhi Centre for Biotechnology, Trivandrum, India, as a postdoctoral fellow during the years 2009 to 2014 at the Department of Neurology, ISMMS, New York, USA, as a postdoctoral fellow in the years 2014 to 2016 at the Department of Neuroscience, ISMMS, New York, USA, and currently working as an assistant professor from 2019 at Nash Family Department of Neuroscience, Icahn School of Medicine at Mount Sinai, ISMMS, New York, USA. On behalf of the Department of Biotechnology, Sree College, Kalady, I'm honored and indeed proud to invite Dr. Merina T. Varghese to this scientific session. Welcome, ma'am.
Thank you,
all.
Madangi. Now, it's time to start our scientific session. After the presentation, we will have time for an interactive session so you can clear your queries by using the chat box facility. Participants, kindly make sure your audio has been muted and your video is turned off. To be inspired is great, but to be an inspiration is an honor. It's my immense pleasure to welcome Dr. Merina T. Varghese, Assistant Professor, Nash Family Department of Neuroscience, Icahn School of Medicine at Mount Sinai, New York, USA, for the scientific session. Ma'am, please.
Thank you, Helen, Sarasena, Layamol, and Matangi for the introductory welcome and the very warm start to this talk. I do not need to repeat what the talk is about since you've already laid out the foundation of what I'll be speaking about, so I'll jump right into it. To outline what I'll be speaking about today, I've divided my presentation into three major sections. The first is a general introduction on neurodegenerative diseases, where I'll touch briefly on amyotrophic lateral sclerosis in particular. And the remaining two sections focus on Parkinson's disease and Alzheimer's disease, which together constitute the two major neurodegenerative disorders known. Also, I've set up a few interactive questions and polls at the link that you see here. You can use the QR code to access it and start putting in your responses. You can also type in the website slido.com and use the hashtag BIOFIESTA 2023 to enter your responses. Please feel free to enter your responses at any time during the presentation. So for the first section, I'll be speaking about neurodegenerative diseases. As you've already mentioned in the introduction, what is neurodegeneration? It refers to the loss of function and the death of neurons in the central or peripheral nervous system. And the clinical features that are associated with neurodegeneration include loss of motor function, sensory function, or cognitive function, depending on what neurons are affected and what brain or spinal cord area or peripheral tissue is affected. The cause, it could be an interaction of both genes and environment, as I will specify for each of the diseases that we talk about today. Single gene causes are rare though. And one example is mutations in the Huntington gene, which cause Huntington's disease. This, in fact, was one of the first known mutations linked to a neurodegenerative disease. However, multiple risk genes are known for many neurodegenerative diseases. For example, in familial Parkinson's disease, as well as in familial Alzheimer's disease, there are many, many risk genes that are known, and many more that are being discovered now as well. Environmental factors can also increase risk. For instance, neurotoxins have been linked to the disease amyotrophic lateral sclerosis. How do we differentiate neurodegenerative diseases? Very often they are divided based on the age at onset into pediatric and adult onset disorders. And I've listed here just a few in each category. So among pediatric disorders, for instance, leukoencephalopathies and epileptic encephalopathies are pretty prevalent. You might have heard of spinal muscular atrophy, and there are several hereditary neuropathies and hereditary pediatric disorders that are neurodegenerative. Among adult onset disorders, which usually show the symptoms once you've reached adulthood, Huntington's disease and amyotrophic lateral sclerosis can develop early in adulthood, whereas dementias, including Alzheimer's disease, as well as motor disorders, including Parkinson's disease, tend to be developed past the age of 50. So they are more age-related neurodegenerative disorders. Let's have a look at what amyotrophic lateral sclerosis is about. You might have heard of this disease in the context of the world-famous physicist who was one of the most well-known sufferers of it, Stephen Hawking. He is one of the rare cases where, after diagnosis, he lived for a long time afterwards, even though his disease was progressing and his cognition was intact. Another well-known personality who had ALS is Lou Gehrig's. Who was a baseball player, very famous in the US. And in the US, amyotrophic lateral sclerosis is also known as Lou Gehrig's disorder. For a brief history, ALS was first identified in 1896 by the French neurologist Jean-Martin Charcot, who is also known as the father of modern neurology. The name itself is derived from a combination of Greek words, A for no, myo for muscle, trophic for nourishment, which taken together describes what happens during the disease. Basically, the muscles lose their nourishment and hence they atrophy. Lateral in the disease name refers to the area of the spinal cord that is affected, and sclerosis is a reference to the hardening of the tissue that accompanies the degenerative changes. ALS is also known as motor neuron disease, and in the US, it's called Lou Gehrig's because of its association with the famous baseball player by that name. The incidence of ALS is most well studied in European populations, and it is known to occur in over 3 people in every 100,000 people, or 1 lakh people. But in South Asia, for instance, the incidence is much lower. It's only about 0.7 cases per 100,000 people. The other thing of note is that in European populations, at least, up to 20% of cases are inherited. So there is quite a strong genetic link for the risks of ALS. What are the clinical features? Mainly, it's the motor symptoms that appear and that cause concern. These involve muscle weakness and difficulty swallowing and speaking, which is often what is first diagnosed. Non-motor symptoms can also develop in up to 50% of patients, and these include cognitive impairment. But of note, it does not happen in all patients. For instance, Stephen Hawking, he was totally able to use his intellect until the end of his life. He was working scientifically, he was publishing, he was speaking, even though it was with the aid of a robotic assistant for speaking. Other features are people lose their capacity for speech, dysarthria. They have difficulty swallowing, dysphagia, and they have behavioral impairments. They also may have muscle cramps, spasticity, and muscle atrophy because of the muscle weakness. What does this look like pathologically? When you look at the gross pathology, what you can observe is atrophy of skeletal muscle as well as the motor cortex in the brain. In the spinal cord, you can see pallor, loss of color, and sclerosis or hardening of corticospinal and corticobulbar tracts. You can also see thinning of the hypoglossal nerves that control the muscles to your tongue, the ventral roots of the spinal cord, and you can see degeneration of upper and lower motor neurons in the spinal cord. Histopathologically, when you look at a tissue section under the microscope and after staining with specific markers, what you can see is aggregation of ubiquitinated protein TDP43, specifically in motor neurons, which you can see here in brown, and the deep brown is the aggregates within the neurons. Sometimes they also form other patterns, like with outside the neurons, but within neurites. You can also see degeneration of upper and lower motor neurons. For instance, here you can see the neurons in blue in a normal, healthy individual. But here in the spinal cord of a patient with ALS, you can see loss of motor neurons. You don't see the cells in blue anymore. You also find diffuse astrogliosis and microglial infiltration in the spinal cord, which is what you see in this area of the picture here. The brown indicates microglial infiltration and astrogliosis within the spinal cord. So what is happening at the molecular level? There are a lot of different pathways involved here. For instance, protein homeostasis, which means the turnover of proteins so that proteins that are abnormal get digested within the cell, and new proteins are made to replace them. That is often impaired. RNA metabolism is also often impaired. The transport within cells, both nucleocytoplasmic and endosomal and vesicular, are often impaired. Axonal structure and function, DNA repair, the release of excess glutamate leading to excitotoxicity, oligodendrocyte degeneration, neuroinflammation, which you saw in the previous slide as astrogliosis and microgliosis, and mitochondrial dysfunction. Many of these are linked to ALS. And as you can see, some of the gene mutations that are seen in ALS are linked to each of these in this diagram here. Of these, I would like to draw your attention to mitochondrial dysfunction over here, which is linked to the SOD1 mutation that's seen in ALS. So many of these features are in fact replicated in the SOD1 transgenic mouse model. And that is what I used in a collaboration when I was a postdoctoral fellow in the Department of Neurology at Mount Sinai Medical School. What we did is to use the SOD1 G93A transgenic mouse, which overexpresses the G93A mutation in the human gene for superoxide dismutase 1. This mouse develops progressive loss of motor function with neuronal loss in the lumbar spinal cord. We double-crossed this mouse so that it overexpresses a protein called PGC1 alpha. Now, PGC1 alpha is a regulator of mitochondrial biogenesis. So if you have more of PGC1 alpha, you should have more of mitochondria and better mitochondrial function. That is our hypothesis, that it would also benefit the mice when they have more mitochondria and better mitochondrial function to overcome the loss that is seen in the presence of the ALS gene. And in fact, we did see better motor function at the post-symptomatic stage, as you can see here. Pre-symptomatic, there wasn't a significant difference. In the mid-symptomatic, you can see that the ALS mice in black here did have a decline in function, but by the post-symptomatic stage, when the ALS mice declined even further, the PGC1 alpha ALS double transgenic mouse was protected. Now you can see there is still a decline as you compare it to the wild type and the PGC1 alpha alone transgenic mouse, but still, as compared to the ALS mouse, it was protected. This is a motor function as measured by the rotor rod task, where the mice have to run on an accelerating wheel, and the ALS mice usually have trouble hanging on to it and fall off quicker than the wild-type mice. That's how we measured motor function. This graph here shows that the SOD1 PGC1 alpha, SOD1 mutant PGC1 alpha double transgenic mouse had greater survival as compared to the SOD1 mutant mouse. Again, showing the protective effect of PGC1 alpha. Even in terms of weight loss, which is quite a significant feature of the mutant mouse, we saw that the weight loss was decreased as compared to the mutant mouse. So it was protected in terms of motor function, survival, and weight loss when we improved mitochondrial function. And what did it do at the histopathological level? We could see improved neuron survival, where when you have loss of motor neurons in the spinal cord of SOD1 G93A mice, the double transgenics were protected, as you can see here in the graph where we quantified the numbers as well. And in terms of mitochondrial function, when we measured the activities of complex 1 of the mitochondrial oxidative phosphorylation pathway, as well as of complex 4 in the spinal cord, we were able to show again that the double transgenic was protected as compared to the single transgenic ALS mouse. This indicates that improving mitochondrial function may be a potential therapeutic mechanism for ALS, at least in the presence of the SOD1 G93A mutation. Now, I spoke about SOD1 so far, but there are at least 4 other major risk genes that are linked to 70% of the familial ALS cases. Note again that not the majority of ALS cases have known mutations, but there are many other risk genes also known in ALS that are seen in 30% of the inherited cases. Other than the inherited factors or the genetic factors, environmental factors have also been linked to ALS. These include, for instance, neurotoxins and cyanotoxins. I leave the question mark here because those studies still need to be validated. Also, athletes have been shown to have a higher incidence of ALS. Reasons unknown. Smoking has also been linked to a higher incidence of ALS. How is ALS diagnosed? Mostly it is based on clinical evaluation. So there is a diagnostic criterion called the L-Escorial and the Early-House criterion, where the physician measures motor function and other non-motor functions that are known to be affected in ALS. Only in familial cases is genetic testing advised, otherwise it is not. There are also some biomarkers that are still at the experimental stage. So for instance, you can measure the levels of certain proteins in the cerebrospinal fluid and the serum levels, and in the serum, the blood serum. These include neurofilament light polypeptide and phosphorylated neurofilament heavy polypeptide, both of which are an indirect measure of neuron loss or neurodegeneration. There's also experimental results showing that MRI of the corticospinal tract and the frontal and temporal regions, as well as the basal ganglia of the brain, can show degeneration. But again, these have not been fully validated for use in the clinic. They are still experimental. What are the current treatments? So far, there are only 2 treatments that have been approved for the clinic. Riluzole, which is a glutamate antagonist. This is supposed to help to reduce the excitotoxicity. Edaveron, which is a free radical scavenger, which targets oxidative stress that is increased in ALS. The other treatments include multidisciplinary care, where you need a team including the neurologist, psychologist, dietitian, speech therapy, a lot of professionals who come together to support the multiple areas that a patient needs help with. There is also symptomatic treatment For instance, for the spasticity, drooling, pain, muscle cramps, dysphagia, dysarthria, among many other symptoms that the patient has to deal with as the disease progresses. There are also many experimental treatments under development. And of these, tofersen, which is an antisense therapy specifically for SOD1 mutations, and Neuron, which is a stem cell therapy, are promising ones to keep an eye on. All right, we shall move now to Parkinson's disease. This is named after the English physician who first described it, James Parkinson. He described it pretty accurately even back then. You can see this description of involuntary tremulous motion with lessened muscular power in parts not in action and even when supported, with a propensity to bend the trunk forward and to pass from a walking to a running pace. You can see that illustrated in this picture of a patient with Parkinson's disease. And very often, one of the earlier symptoms is, in fact, that the patient has trouble moving or initiating movement. And once they start moving, they don't have the ability to control the pace of the movement. It can go from very slow to very fast. And they have tremors when they're not doing anything. And even if they support their arm, sometimes the tremor and the pill rolling movement of their fingers continues. The incidence of Parkinson's indicates that it's the second most common neurodegenerative disorder, with Alzheimer's disease being the first. And it tends to increase in incidence with age. So you can see past the age of 50, the incidence shoots up pretty rapidly. And if it's corrected for person years, for age of the person, you can also see that the incidence is much higher in men than in women. Among the clinical features, the cardinal features that are used for diagnosis are a combination of motor symptoms, which includes bradykinesia, which is the movement, the difficulty to initiate movement and the difficulty to control the pace of movement that I just described in the earlier slide, together with the presence of one of either rigidity or tremor. So it's a combination of these clinical features that is used for diagnosis. However, there are also many non-motor symptoms. So you can see here that even though the hallmark for the diagnosis, bradykinesia, rigidity, and tremor occur here, many of the non-motor symptoms occur much, much earlier, before the motor symptoms start to even be detected. So it can start from sleep disruptions and behavioral disorders, depression, constipation, anxiety, hyposmia, which is decreased sensation of smell, excessive daytime sleepiness. All these can happen years before any of the non-motor symptoms are even appearing. And as the disease progresses, even other symptoms can appear, like cognitive impairment, loss of urinary control, dementia towards later stages, together with increasing progression of the motor symptoms, including dyskinesias, dysphagia, so difficulty swallowing again, postural instability and gait disorders and falls. What are the pathological features? Here, there's a clear link between loss of dopaminergic neurons in the substantia nigra and the motor functions that you see in the disease. So as you can see here in the control brain, the substantia nigra has most of its dopaminergic neurons, which are seen here in deep color because of their neuromelanin pigment. But in a Parkinson's disease patient, you see a certain specific region, the dorsolateral component of the substantia nigra, loses a lot of the dopaminergic neurons. Another feature often seen is that this loss is hemispheric, so you have it only on one side, so it's unilateral, which means that the motor symptoms also appear on one side first, usually on the opposite side of your body. And there is also a progressive spread of aggregated alpha-synuclein inclusions, which you see here in deep brown. And here You can see the morphology of them, which they're like dense inclusions within the cell body of a neuron, or they can be even in the processes, the neurites of the neuron, or they can be in both the cell body as well as in the neurites. And usually this aggregation of alpha-synuclein protein tends to occur in a very progressive manner as the disease stage progresses. So at the earliest stages, it's seen only in certain areas of the basal ganglia. And as the disease progresses, the pathology also progresses from Braak stages 1 through 6. What happens at the molecular level here? Here again, you see a pattern. You'll see it emerging among many neurodegenerative disorders. In fact, you have a defect of proteostasis, in this case linked to alpha-synuclein. You have a dysfunction of mitochondria, oxidative stress, calcium homeostasis, axonal transport, neuroinflammation, which you can see many of these connect to each other. The mitochondrial dysfunction, for instance, induces the oxidative stress, which in turn can affect the proteostasis of alpha-synuclein, which in turn can affect other pathways such as the vesicular and lysosomal degradation pathways. And all of these can be played into also with activated microglia, which secrete factors and in turn influence processes within neurons and other cells around them. And all of these terminate in cell death. Another interesting hypothesis is that the aggregated alpha-synuclein can be released by dying neurons, and it can spread to unaffected neurons in the vicinity. This again is a pattern that emerges in other neurodegenerative disorders and is sometimes referred to as the prion mode of spreading. You might have heard of infectious protein particles or protein inclusions called prions linked with mad cow disease, for instance. So that's also an ongoing hypothesis in many areas of neurodegeneration. I'd like to draw your attention again to mitochondrial dysfunction, which is an area that I focused on during my PhD studies with Dr. Kochipurikal Mohanakumar in IICB in Calcutta. There, we found that there's decreased complex 1 activity in blood platelets from Parkinson's disease compared to controls. So you might remember the oxidative phosphorylation pathway from your biochemistry lessons, where in mitochondria, electrons are passed across complex 1 through 5 and then to ATP 1 through 4 and then to ATP synthase to generate ATP. And it is complex 1 specifically that is affected in Parkinson's disease as compared to controls, not complex 2, as you can see here. Given this information, we developed a cytoplasmic hybrid or a cybrid model where we fused blood platelets to a cell line that was deprived of its mitochondrial DNA. And this cell line was a neuroblastoma cell line derived from a human. So we could, in fact, differentiate the cybrids into neuron-like cells. And this was work done with Ambali Appukuttan in the lab, who is also a student who graduated from Cochin University. She and I differentiated PD cybrids and showed that when they developed from cell lines to neuron-like cells, the neurons that they produced showed decreased length and complexity of their neurites, as you can see here and here. And also, they were less viable than controls, which means that the cells died faster. The PD cybrids also released less dopamine, as you see here, and they expressed less tyrosine hydroxylase compared to the parent cell line. Now, tyrosine hydroxylase is the enzyme that produces dopamine. So decrease in its levels can be linked to the decrease in the dopamine secretion as well. So here we were able to replicate by transferring mitochondrial DNA from PD patients into a cell line. We were able to replicate some of the features that are also seen in the brain of Parkinson's disease patients. Now, what are the genetic and environmental contributors to Parkinson's disease? There are many monogenic mutations that cause inherited PD, but this is again only in 5 to 10% of cases that are familial. I've listed here a few of the genes and the proteins that they encode, and you can see that synuclein, which we talked about in regards to the protein inclusions that show up in the pathology, is one of the genes that is mutated. There are also several identified risk genes, such as glucocerebrosidase, which is one of the most common genetic risk factors. And there have been links that are been found between organic pollutants and pesticides and an increased risk of Parkinson's disease. How is PD diagnosed? In addition to the clinical diagnosis, there's also imaging biomarkers that can be used to detect the decline of dopamine in the brain by F-dopa PET. You can also look at SPECT to see decreases in the SPECT signal. You can also use MRI to see decreased binding of the dopamine. There are also biomarkers that are used, that are being developed to look for cerebrospinal fluid and blood levels of different proteins. And there are also screening for motor or non-motor symptoms using digital tools. So for instance, you can look for those early signs of sleep disruption or a loss of smell, things like that. You can try to link those with a later PD diagnosis. And those are still, again, experimental, but very promising. What are the treatments? So the best known treatment is L-dopa, which directly substitutes the precursor of dopamine. And it restores striatal dopamine, which goes down because of the loss of dopaminergic neurons. There is also medication for many of the motor and non-motor symptoms. So that includes selective serotonin reuptake inhibitors for the psychiatric symptoms, cholinesterase inhibitors for cognition. And in addition to supplementing the striatal dopamine, some of the other enzymes in the pathway can be inhibited so that dopamine is not used up, or L-dopa is more preferentially converted to dopamine. And also, you can inhibit MAO-B, which again prevents the recycling of dopamine. You can have dopamine agonists, which act like dopamine at the receptor. So there are many ways in which the dopaminergic pathway itself can be targeted. In addition to pharmacological treatments or medications, there is also deep brain stimulation, where an electrode is implanted into one of the dopaminergic centers of the brain, and the clinician can apply an electrical stimulus to stimulate those neurons and stop the tremors and the motor dysfunction. This has also been found to be extremely effective, especially in patients who are responsive to L-dopa. Also, rehabilitative therapy and exercise have been shown to help patients. And in experimental treatments, we have gene and cell-based substitution of striatal dopamine, which is being investigated, as well as clearing and transport of alpha-synuclein aggregates. All right. So now we can move on to the third section and the final section of my talk, which is on Alzheimer's disease. Here, as a brief history, it was described by Alois Alzheimer, who's a German psychiatrist and neuropathologist. He described the clinical and pathological features in his patient, Auguste Dieter, who was a 51-year-old woman. So it was a case of early onset dementia. Now, even though dementia has been mentioned in the literature much earlier than Alois Alzheimer's description, what is key is that he linked the clinical features of dementia with the pathological features that are now known to be because of pathological aggregations of protein. So he was the first to do that. And that is what made his the first definitive description of the disease. Now, something to note is that though it is the most common neurodegenerative disorder, 60 to 90% of dementias are explained by a diagnosis of Alzheimer's disease, but that does not exclude other causes of dementia as well. So you can see here that the autopsy confirmed Alzheimer's disease is in orange here, but there are many other diseases that contribute to dementia linked to aging. For instance, cerebrovascular disease, hippocampal sclerosis, frontotemporal dementia. There's also dementia with Lewy bodies. Lewy bodies being alpha-synuclein aggregates similar to the ones that you see in Parkinson's disease. So there are many other causes that can lead to dementia. And all-cause dementia at 65 to 70 years is 1 in 100 per year, and it increases even further past the age of 80. It becomes 4 in 100 people per year. What are the clinical features of Alzheimer's disease? Mainly cognitive decline in combination with difficulty with language and difficulty in spatial orientation. So I'm sure many of you have heard of stories where older folks have gotten lost, not able to find their way home, and that's often when they get a diagnosis of Alzheimer's disease or are taken to a neurologist at that stage. Pathological features include at the gross pathology level, if you compare the brain from a healthy aged person with that of a person from Alzheimer's disease, and these two brains are from two men who are exactly the same age, 85 years of age. You can see the brain from the man with Alzheimer's disease has shrunk in comparison to the brain from the healthy aged person. And you can also see that there is a widening of the sulci, the grooves between the mounds in your brain, which indicates that there's a loss of tissue, basically. If you take a coronal section of the brain in this angle and look at it, comparing a healthy aged to an Alzheimer's disease brain, again, you can see evidence of the overall shrinkage, as well as certain particular areas being affected more. For instance, the temporal lobe here, the hippocampus, the lateral ventricle, which is enlarged, and the hippocampus and the temporal lobe that are shrunk. All of this is associated with pathology that you can see under the microscope. So the hallmark features are extracellular amyloid beta plaques, which you see here as diffuse A beta plaques, or as cored A beta plaques, which have a dense center and a halo-like surrounding. You also see intracellular neurofibrillary tangles of the microtubule-associated protein tau, which you see here as tangles, as well as what they're called as ghost cells or ghost tangles, because you can no longer see the nucleus of the cell. You can also see them in the neurites or the processes of neurons, and you can also see them associated with plaques and around them, and which are referred to as neurotic plaques. Alongside these, you see dystrophic neurons or neuronal death. You see inflammation, again, a common theme. In this case, microglia as well as astrocytes are involved in the neuroinflammation. You see cerebral amyloid angiopathy, which is a deposition of amyloid beta in the blood vessels. You also see oxidative stress. And this leads me into what happens at the molecular level. Mainly, a lot of the studies have focused on amyloid beta and tau pathology, which again tend to follow a very progressive pattern. So A beta, for instance, when there's no dementia, you see hardly any. But when you have pre-Alzheimer's disease or non-Alzheimer's disease dementia, you tend to see a lot of A beta plaques in the neocortex in phase 1. In phase 2, it also spreads to the allocortex, which includes the hippocampus and the entorhinal cortex. And in phase 3, it spreads to the striatal areas, which you heard about in relation to Parkinson's disease as well as in the subpial and post-cingulate gyrus over here. In symptomatic AD, in phase 4 and 5, it spreads even further to the midbrain, the substantia nigra, which you remember from Parkinson's disease. the cerebellum, and the reticular formation of the pons. In contrast, the neurofibrillary tangle stages show a different pattern. So the earliest neurofibrillary tangles appear in the locus ceruleus, but this, however, is not linked to Alzheimer's disease. It happens even in the 10th decade of, in the 10 to 20 years of age. And in normal aging stages 1 to 2, again, it appears mostly in the entorhinal cortex region. However, in Alzheimer's disease, it spreads further afield. So it goes into the neocortex, it goes into the hippocampus, and at much later stages, it spreads almost throughout. Typically, they start in the axons of the neurons and kind of spread towards the soma. So as the disease progresses, you'll see it in neurofibrillary tangles within the neuronal cell body. And at the very late stage, it affects almost all areas of the cortex, including the association cortex, as well as the sensory cortices. In addition to amyloid beta and tau pathology, apolipoprotein E is a major risk factor of Alzheimer's disease, and other molecular features are loss of synapses, dysfunctional proteostasis, again a repeating theme, mitochondrial dysfunction, again, and neuroinflammation. So this leads to some of my current work as a faculty in the Department of Neuroscience, together with my mentor, Dr. Hoff. We are looking at brain region-specific vulnerability of neurons in Alzheimer's disease using a technique called multiplexed immunofluorescence. So if I can direct your attention to the bottom area of the slide here, we start with human postmortem brain samples from the prefrontal cortex and the primary visual, embed them in paraffin, section them, stain them, and then put them on a slide as a tissue microarray. We stain them with multiplexed immunofluorescence for 23, now it's 40 markers, including markers for various neuronal types, for Alzheimer's disease pathology, including A beta and tau, for various kinds of non-neuronal cells, including glia and vascular cell types. And then we did an image analysis so that we could detect all the various cell types in relation to the pathology. And our early results show that the prefrontal cortex neurons, when you compare controls with early Alzheimer's or mild cognitive impairment and late stage Alzheimer's disease, prefrontal cortical neurons are much more affected than neurons in the primary visual cortex, where it's only at the later stages of Alzheimer's disease that you see such a decline. What happens at the levels of genes and environment here? Many of the dominantly inherited rare mutations, including mutations in APP, presenilin 1 and 2, have been linked to familial Alzheimer's disease. But the strongest risk factor is the apolipoprotein E gene, in which the epsilon 4 variant increases the risk of Alzheimer's disease. However, there are more than 600 other genes that have been linked to increased risk of Alzheimer's disease. And the important point to note is that many of these seem to be acting in combination. So they might not be acting just one mutation at a time. In addition to the genetic factors, there are many lifestyle factors that can also contribute, including, for instance, metabolic factors such as diabetes, hypertension, obesity, and low HDL cholesterol in midlife, as well as hearing loss and traumatic brain injury. In later life, smoking, depression, low physical activity, social isolation, diabetes, and air pollution are risk factors. What are the diagnostic biomarkers? Currently, PET using fluorodeoxyglucose and magnetic resonance imaging or MRI and CT or computed tomography to detect the structural loss of neurons are used as biomarkers. There are also experimental biomarkers from the cerebrospinal fluid and blood, such as the levels of the A beta 42 protein in the cerebrospinal fluid and the CSF tau protein levels, which are used as experimental or research biomarkers. And screening for related symptoms is also being investigated as a biomarker. Note here that there is a very well-known temporal progression of the various biomarkers. So A-beta 42 in the CSF is one of the first to appear, and the neuronal loss is detected much later. So before the clinical symptoms occur, you can actually detect the biomarkers. And this is important because once neurodegeneration sets in, it's difficult to reverse. So if you can catch it before when the biomarkers happen, that might be a better time to treat. And what are the treatments? Currently, it's cholinesterase inhibitors that are available for clinical therapy, as well as an NMDA receptor antagonist. And the other treatments are all for comorbidities such as depression and anxiety. Among the experimental treatments, recently, aducanumab and lecanemab were approved by the FDA, and these are both anti-A-beta targeting antibodies. Now, what's to note is that this one, even though it reduced the A-beta levels, did not do much in terms of cognitive, holding back the cognitive decline, versus this, which did not target A-beta plaques like aducanumab, but targeted A-beta oligomers, seems to be more promising in terms of helping with cognitive decline. So this is again showing that targeting the therapies at various stages of the disease progression is important. So to summarize, for all the neurodegenerative diseases that we've discussed today, and this is just a small view of 3 of many, you would have noticed that the clinical features are linked to which specific neurons and which brain or spinal cord areas are affected. The disease pathology often involves progressive protein aggregation. There are many shared pathogenic features like mitochondrial dysfunction, neuroinflammation. Genetics and environment interact in the disease onset. There are many biomarkers already available and many under development. And the existing treatments are mostly symptomatic. So there is a huge need for cures, which is where most of our work is focused. With that, I would like to say thank you and acknowledge the many, many people who contributed towards these studies from the various groups that I've worked in over the years, as well as my collaborators who are involved in the current studies in my lab. Thank you for your attention, and I would like to open the floor for questions.
Thank you, ma'am, for giving such an informative session. Now it's time for the interactive session, and I request the participants to come forward and put their queries in the chat box.
Ma'am, can I ask you a doubt?
Yes, go for it.
Am I audible now? I'm having
Yes,
some
you are.
network issues. Yeah. Ma'am, in some case of Alzheimer's disease and all, doctors or the clinician usually prescribe donanemab. It's a tablet that's contain donepezil and memantine. So how it actually acts on the brain cells, ma'am, to inhibit the intensity or prognosis of the Alzheimer's disease, ma'am?
That's a good question. And I can point you towards the information on this slide. So donepezil is a cholinesterase inhibitor. And one of the early pathways that was discovered to be affected in Alzheimer's disease is a loss of acetylcholine function. And that is what donepezil targets. It tries to stop the loss of acetylcholine function, basically.
Okay.
And memantine, when used in combination, acts as an NMDA receptor antagonist. So it counteracts the effect of glutamate at its receptors. NMDA receptors are a subtype of glutamate receptors.
Okay.
So what it's trying to do is to change the activity of two different neurotransmitter systems that are known to be affected in Alzheimer's disease.
Okay.
I hope that answers your question.
Yes, ma'am. Yes. Thank you.
Yeah, you're welcome. Now note that neither of these treatments is a cure. They help to slow the progression of the disease to some extent,
Yes,
but it is not a cure by any chance.
Okay. Yeah.
Ma'am, there are some questions in the chat box. May I read out?
Yes, please.
Okay, first question. Ma'am, what are the recent advancements of nanomedicine in neurodegenerative diseases?
Could you be a bit more specific as to what you mean by nanomedicine? Because I think it could mean several different things, and I'm not sure what exactly you're asking.
Ma'am, can I interrupt?
Yes, go ahead.
Yeah, ma'am, I think they need to know if there are any advances in treatment in the scenario of if we can incorporate nanomedicine in the application of neurodegenerative disease, something like that they were asking. We have some conventional neurodegenerative disease treatments we have. So if there is any application in related with nanomedicine, that's what we wanted to know. So in cancer treatment and all, we are using gold nanoparticles for the active target specific drug delivery and all. No, so they wanted to know whether is there any nanomedicine application in the case of neurodegenerative disease, something like that.
I can't put my finger on any one that comes to mind right away, really. I know that there are drug delivery mechanisms being investigated, so that's potentially one area that could be helpful in neurodegeneration as well, because, you know, getting things into the brain is the first, because of the blood-brain barrier, getting any medication into the brain is the first thing that you need to consider for any neurodegenerative disease.
Yeah.
So for that, nanoparticles could be helpful. I can't pinpoint one particular example for you at this point. I'd have to go back and check for that.
No problem. Thank you.
Yeah.
Ma'am, next question. Could you please tell us how prenatal diagnosis of neurodegenerative disease is done and to what extent it is effective?
So this would be a question more in the area of pediatric disorders than for age-related neurodegeneration at this point, because at least for now, not much has been done in terms of very early neurodevelopmental links to neurodegeneration. So in the area of pediatric neurodegenerative disorders, there are some that have a very well-known hereditary link. So the genes are well known, and those can in fact be diagnosed by genetic testing. Some of these are very rare disorders and are known to be more prevalent in certain populations. So they might be advised to have genetic screening specifically for those diseases. And some of them are, because they're familial, and if there's a family history, then the physician will, again, advise for prenatal screening. Some of the genetic disorders in pediatric conditions, once they are diagnosed, can actually be treated, some of them by dietary interventions and such. So in those cases, in fact, it's very helpful to have a prenatal diagnosis and very early treatment.
Thank you, ma'am. Next question. Ma'am, could you please explain the function of alpha-synuclein?
That is a good one. Well, the interesting thing about many of these proteins that aggregate, including alpha-synuclein, in fact, is that their normal physiological function is still being investigated. So alpha-synuclein, for instance, it's known to be present in axons. So this possibly an axonal function there. But beyond that, I don't know really. And yeah, I mean, I can't answer the question because I don't know beyond that.
Okay, ma'am. Next question. How can we reduce the risk of neurodegenerative disease?
So for the most common two, for Alzheimer's disease and Parkinson's disease, Alzheimer's disease to some extent, because there are links to like your lifestyle, taking care of your cardiovascular risk is a very good way of reducing your risk for Alzheimer's in later life. So if you have a family history of diabetes, hypertension, high blood cholesterol, manage those using medications, get checked regularly and manage those. Those will in fact help to reduce your risk of Alzheimer's disease. And also, like if you notice hearing loss in your middle age, get that checked out. As you can see in later life, some of the risk factors are low physical activity and social isolation, which was a huge thing during COVID. So in fact, the incidence of Alzheimer's disease seems to have increased after COVID in the older population. So those are things that could be you could intervene. You can make sure that you stay physically active and that you mingle with friends and family so that you can prevent, at least to some extent, your risk for developing Alzheimer's disease. So right now, that's the best advice we can give you for Alzheimer's. For Parkinson's, again, there is a treatment available, like not a cure, but if you follow up whenever you notice any problems like issues with sleep, with issues with loss of smell, early motor symptoms, go to a neurologist, get it checked out so that you know whether you have a tendency or a family history of Parkinson's and can start treatment early. Because many patients are responsive to L-DOPA. And again, there is the surgical intervention as well. I hope that answers your question.
Yes, ma'am. Thank you. Next question. Is brain implants helpful as a therapy to neurodegenerative diseases?
Yes, at least for Parkinson's disease. So when you talk about brain implants, it can mean several different things. One of them is implanting electrodes, where it's kind of like you have a cardiac pacemaker, but in this case, it's a brain pacemaker. So you would be electrically stimulating the cells that are, or the circuits that are missing. So that is effective in neurodegenerative diseases where you know a particular brain circuit is affected and where that brain circuit is very, very regionally restricted. So that's the case in Parkinson's disease, for instance. But in something like Alzheimer's disease, where you have so many brain networks affected, it's extremely difficult to do an implant in one area and then help to treat the disease. But other things that people are also looking at are implanting stem cells, for instance. So the idea there being that the implanted cells then go and make connections where the connections are lost. This is still very, very early stage, of course. So there's years to go before we can say for sure whether that works or not. This could be promising, for instance, in diseases where you have loss of spinal cord neurons and where you need to reconnect the motor neurons. Another thing that could be implanted is the genes themselves. So that's, again, an area that's being investigated. It's not ready for the clinic yet, but definitely, especially in neurodegenerative disorders where you have a single gene mutation and you know what the mutation is, the particular mutation in that patient is, you may be able to correct it if the mutation is a loss of function by providing the corrected gene in the cells that need the protein to be expressed. So at least 3 different kinds of implants that I can think of that could potentially be good treatments for neurodegenerative disorders.
Yes, ma'am. Next question. Ma'am, what were the major risks you faced while working on PGC1 alpha gene?
The major risks that I faced. I am not sure I understand the question completely, whether it means as a biosafety risk in the lab or whether it means like an issue with the mice themselves. But I'll try to address both. So for the transgenic mice, what we noticed is that the protective effect was only present if PGC1 alpha was expressed specifically in neurons. If PGC1 alpha was, the expression was driven by a promoter like actin, which expresses in most cell types, we did not observe the same level of protection. So it appears to be a very neuron-specific protective mechanism. So that's something that would need to be considered if you're designing a drug that would have a similar effect. So that's the risk at the level of developing the therapy. At the level of the researcher, I mean, beyond the safety precautions that you would take working with animals, that was about it, really.
Yes, ma'am. Next question. Difference between Alzheimer's and senile dementia.
So senile dementia was one of the earlier terms that was used for Alzheimer's disease before the pathological features were well defined. So at that time, it was used kind of interchangeably. So when you read the older papers, sometimes both terms are used interchangeably. What you'll need to do when you're reading papers that say senile dementia is to see whether they also talk about Alzheimer's disease pathology. So you'd have to see whether they have a description of plaques and tangles and whether they have, at least after the proteins were discovered, whether they also talk about A beta plaques and tau tangles or not. And otherwise, it could be other kinds of dementia too. So that's a distinction you'll need to make when you're reading the literature, whether it's actually Alzheimer's type dementia or dementia due to old age, which is what senile refers to really.
Okay, ma'am. Next question. How developed is the researchers based on neurodegenerative disease currently?
I am a little confused by the question. So how developed are the researchers? I guess it depends on what you're looking at, really. So when you're studying neurodegeneration, you can look at things at the clinical level. So, there are, for instance, there are things that you can do to improve clinical diagnosis. That can be even something as simple as translating an existing diagnostic criterion into a native language. That can be something really important because sometimes some of the criteria that are used for diagnosis are very, very region specific. So some terms that are used may not translate well to another part of the world. But if you know what is being diagnosed and why they're asking those questions, you'll be able to translate it so that it's appropriate for another geographic context. So it could be something as simple or what seems as simple as that to something as complex as trying to design a genetic intervention. So yeah, the advances can be at many different levels. I hope that kind of answers the question because I wasn't sure what the questioner meant.
Okay, ma'am. Next question. How are biomarkers used in disease diagnosis and why are new biomarkers needed for neurological diseases?
Right. So biomarkers can refer to a wide variety of things. Usually what it means is something that helps you to say if a person has X, Y, and Z, then the person has disease A, B, or C, right? So for instance, for diabetes, it's pretty easy. Like if you have a blood glucose level that's above a certain range, and if you have an insulin level that's below a certain range, you know, okay, this person is pre-diabetic or he or she is diabetic, right? But unfortunately, for many neurodegenerative diseases, you don't have a biomarker that is as clear cut. So some diseases do. For instance, in Parkinson's disease, we know exactly which neuronal cell types are affected. So if you see that those neurons are lost when you do an MRI, for instance, or when you do an L-DOPA PET, which specifically looks at the levels of L-DOPA in that brain area, then you know, okay, this is most likely, or this is very highly likely to be a case of Parkinson's disease. But for something like Alzheimer's, the comparable marker in PET would be something that can detect amyloid beta or tau. Now, the problem is amyloid beta and tau are also seen in normal aging. The thing is, where they are seen, when they are seen, and at what levels they are seen makes a difference as to whether the person is on the trajectory of Alzheimer's disease or whether it's a case of just normal aging without any cognitive loss. So that makes it extremely difficult to use A beta or tau as biomarkers for Alzheimer's disease because they're also seen in normal aging, right? So what people are trying to do is to look at specific types of A beta and specific types of tau in the blood or in the cerebrospinal fluid, which are more accessible than a person's brain when they're living, to see whether they can diagnose Alzheimer's disease separately from normal aging. And that's an example of why you need newer and better biomarkers.
Thank you, ma'am. Next question. Risk of neurological diseases before and after the COVID pandemic.
Good one. I mean, unfortunately, the COVID pandemic seems to have increased people's risks for a lot of both psychiatric and neurological disorders. And we don't know exactly why yet. We do know that people have been reporting increased incidence of mental health issues as well as neurological symptoms. I mean, everyone knows, for instance, about the loss of smell and the change of smell and taste after COVID. And that points towards a neurological condition, really. So long term, in terms of neurodegeneration, we don't know yet because a lot of these diseases take decades to develop. So we'll be knowing about what the long-term effects are 10, 20, 30 years out from now, really. But there are a lot of people already looking at what COVID-19 has done to people's brains. So in fact, like brain banks, like at the place where I work, the hospitals have been collecting tissue from like brains from people who died of COVID so that we can try and understand what's going on at the neurodegeneration level.
Okay, ma'am. Next question. Can CRISPR cure Parkinson's?
Ooh. That is assuming that Parkinson's has a single gene cause, right? So first of all, there's a minority of Parkinson's disease cases that have a single gene cause. So that makes it pretty difficult to begin with. And even in those cases that have a single gene cause, you're assuming that the cause is due to a loss of function. Oftentimes what happens is the mutations also can cause a gain of function, and it can be a gain of toxic function. So synuclein, for instance, it might be losing its normal physiological function and taking on a toxic function that leads it to aggregate and become neurotoxic. So in cases like that, cutting out the gene or replacing it by CRISPR may not do what you want it to do in terms of curing the disease. But in cases where you have a loss of function for the gene, potentially you can use CRISPR as a therapy. Again, remember, it's easier to do if it's a very targeted brain area or a very targeted CNS area that's affected, really. It's more difficult if it's like throughout the brain or a very, very wide area that you need to target with gene therapy.
Okay, miss. Next question. Role of gene therapy in Parkinson's.
I think that kind of ties to the earlier question, even though they're not overlapping. Gene therapy is being tested in some animal models, at least mouse models of Parkinson's disease that are driven by some of the Parkinson's disease mutations, using viral vectors, for instance, to deliver the normal non-mutated genes. So, and because it is specifically in dopaminergic cells, possibly it could be treated if you intervene early enough before you lose the majority of the dopaminergic neurons. So potentially in familial cases with a known family history and where the person is carrying the risk gene, it maybe could be a therapy.
Okay, ma'am. Next question. What are the neurotransmitters affected in Parkinson's other than dopamine?
So the major neurotransmitter that's affected is dopamine, but then towards later stages, you do have cognitive involvement, which suggests that you have the acetylcholine system, the cholinergic system also affected. You also have depressions on the psychiatric side, like depression, sleep disorders, which point towards probably a serotoninergic involvement as well. So multiple pathways can be affected at various stages of the disease as it progresses.
Okay, ma'am. Next question. It's often seen that men are most likely to develop Parkinson's disease than women. And why is that so? Is there any scientific reason?
There are reasons under investigation, but it's not fully known why. So the interesting thing is it's not just in Parkinson's disease, it's also in Alzheimer's disease. There are certain populations where, because women are protected, people are looking at whether it's because of a hormonal protection effect. It could also be in Alzheimer's, it can flip the other way because women tend to live longer. They might be diagnosed with Alzheimer's because they live longer and hence have an increased risk, because age is the greatest risk factor for Alzheimer's, really. So it can be because of hormonal factors that we are still looking into, but yeah, still an open question for research.
Ma'am,
Yes.
I have a doubt, ma'am.
Yeah, go for it.
Ma'am, the onset of these neurodegenerative diseases increasing in these decades. So is there any prevention or any antioxidants, the intake of any antioxidants can prevent or lesser the extent of this onset, ma'am?
So far, there haven't been any that have been proven clinically, and several have been tested. Starting from curcumin to many other antioxidants, even like mitochondrially targeted antioxidants have been tested. At least preclinically, nothing has been proven to be protective yet. That said, nothing has been shown to be harmful either. So incorporating some of them into your diet may not be a bad thing either.
Okay, thank you, ma'am.
Yeah.
Good morning, ma'am.
Yes.
Okay. Actually, ma'am, I just have a doubt that you said somewhere in your, initial section that an inclusion of a single transgene was enough to restore the functions of mitochondria through
Yes.
single or a double cross, and it's in the that effectiveness was more when it was double crossed. My
That
question is, ma'am,
yeah.
my question is that a particular gene codes for any regulatory molecule that is involved in the control of the expression of certain other genes that are usually meant for usually code for the structural components of the mitochondria, or how exactly this particular molecule interact with other genes to control them, or I just want to know the molecular mechanism behind this particular gene, means
Yeah.
this treatment.
Yeah. So this gene, PGC1 alpha, is peroxisome proliferator gamma coactivator 1 alpha in full. It is a regulator of the biogenesis of mitochondria. So it does increase the number of mitochondria. It drives the structural components of mitochondria and hence increases the number of mitochondria. It may also have regulatory effects on other genes. So I haven't looked into that in detail. For this particular study, we were looking at the mitochondrial function because it's known to be affected in the ALS mice, but it's possible it could be affecting other pathways as well.
Ma'am, biogenesis genesis of mitochondria means there must be some critical factors actually affecting after the inclusion of this particular gene.
It could be everything from mitochondrial DNA replication to the structural proteins being translated. So there are multiple things where it could be affecting. Yeah.
Yeah, it is. That it's quite fascinating actually. Again, one more question is: so far, that much biomarkers are not available for the early diagnosis of the conditions like AD and PD. Could you please suggest any methods that can be explored well enough to screen those biomarkers if we have the samples from the patients who is suffering from this kind of diseases?
Yes. So that's a multi-layered question, actually. So the first layer is how you define your patient population. In research studies, like the stratification of your patient population is extremely important because for something like Alzheimer's disease, for instance, it could overlap with so many other different kinds of dementia, right? So you need to have a combination of clinical diagnosis and, if possible, scanning like PET or MRI diagnosis, and also, if possible, blood or cerebrospinal fluid biomarkers. Now, among the blood and cerebrospinal fluid biomarkers, there are many that are being tested and many different methods that are being tested as well. A lot of the methods are antibody-based, so your detection and sensitivity is as specific as your antibody binding is to the particular subtype of your protein and the particular aggregation state of your protein. So all of that can play into how accurate your biomarker development is. And a lot of what biomarker development is about is being able to tell with sensitivity as well as specificity whether the diagnosis of whichever disease you're looking at matches with the levels of the biomarkers that you're seeing or the ratios of the different biomarkers that you're looking at. I hope that addresses your question fully.
Yeah, I guess so. Anyway, thank you. That was a nice presentation, and as a beginner, I think this will help me a lot in future. Thank you.
I'm glad. Yeah.
Thank you, ma'am. I think almost all the queries were cleared by the speaker. Now it's time to wind up the session. Gratitude is a powerful catalyst for happiness, and a grateful heart is a magnet for miracles. Now I invite Elsa for the vote of thanks.
It is my great privilege to give a vote of thanks on this occasion of our international webinar series, BIOFIESTA 2023. On behalf of the Department of Biotechnology, I would like to extend my heartfelt gratitude to our esteemed resource person, Dr. Merina T. Varghese, for her talk, which was very informative and interesting. The webinar provided an inclusive insight into the importance of research in neurodegenerative diseases and their various aspects. Thank you once again, ma'am, for the well-structured and knowledgeable presentation. I take this opportunity to express my sincere gratitude to our principal, Dr. Preeti Nair, the management of Adi Sankara Group of Institutions and IQAC for the encouragement and strong support rendered in the successful conduct of this webinar. Last but not least, I would like to thank the faculty members, other participants, and all of you present here for making the time to be with us today and helping us make this event a grand success. Once again, thank you all.
Thank you, Elsa. The link for the feedback form is provided in the chat box and it will be active only for an hour after the session. Thank you all for being a fabulous audience and supporting us with your cooperation. The next webinar on this series will be coming soon. Once again, thank you all and wishing you a wonderful day ahead. Thank you.
Thanks all.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
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 Thank you, ma'am.